Hello everyone, I'm Xiaohong Liang from Department of Immunology, Shandong University. Today we are going to talk about immunotherapy for tumors. From immunoediting concept, we already know finally tumor escapes from immunosurveillance. Given that multiple pathways of suppression are at play in tumor microenvironments, including suppressive immune cells, as well as molecules such as immune checkpoints. Thus, pharmacological blockade of these inhibitory pathways and to boost anti-cancer immune response are thought to be potent therapeutic strategies for tumor. In general, we classify the anti-tumor immunotherapy into two types. The first class of strategies is to stimulate the active anti-tumor immune responses by using vaccines, cytokines, or by blocking immune checkpoints. The second class is to passively transfer anti-tumor antibodies or immune cells. Let's look at the first class of anti-tumor immunotherapy. Vaccination with tumor antigens is performed to actively stimulate anti-tumor immune response. This picture shows the history for the identification of important tumor antigens and the application of tumor vaccines for therapy. We can see that there is a long history for tumor antigen identification and tumor vaccine application. According to the form of tumor vaccines, they are classified into several types, including killed tumor vaccine, purified tumor antigens, dendritic cell-based vaccines, cytokine or co-stimulator enhanced vaccines, DNA vaccines, or viral vector-based vaccines. Among of them, DC-based vaccines are one of the most promising types. Sipuleucel T is one of successful DC-based tumor vaccines, which is stimulated by prostatic acid phosphatase (PAP) and approved by FDA in 2010 for the treatment of asymptomatic or minimally symptomatic metastatic resistant prostate cancer. This slide shows the effect of tumor vaccines on anti-tumor immune response. They induce to generate new antigen-specific T-cell responses against tumor. Secondly, cancer vaccines can also amplify existing tumor-specific T-cell responses. Finally, cancer vaccines can increase the breadth and the diversity of the tumor-specific T-cell response. Together, these effects can result in regression of tumors. The second active strategy is to augment host immunity to tumors with co-stimulators and cytokines. Co-stimulatory genes like B7 or cytokine genes like IL-2 could be transfected into tumor cells. Then, these tumor vaccines with expression of co-stimulatory genes would activate tumor-specific T cells. In addition, commercially cytokine products such as IL-2 interferon gamma could also be systematically administered, but the side effects is serious. The third strategy for actively stimulating anti-tumor responses is to block immune checkpoints on immune cells. Then, what are immune checkpoints? Immune checkpoints are molecules in the immune system that turn down the activating signaling and inhibit immune response. They were also called the brake of immune cells. Thus, the specific antibodies or inhibitors against immune checkpoints are developed and used to release the brake on immune cells. As a result, the immune cells will be efficiently activated and induce strong anti-tumor response. This picture shows that immune checkpoint therapy significantly inhibits tumor growth in mice. Excitingly, in tumor patients, immune checkpoint inhibitors also show surprisingly efficacy. Apart from active immunotherapy, immune cells or tumor-specific antibodies could be adoptively transferred or injected to the patients. Firstly, let's look at the adoptive cellular therapy, including tumor-infiltrating lymphocytes, tumor antigen-specific T lymphocytes, NK cells, and NKT cells. Tumor-infiltrating lymphocytes could be isolated from tumor tissues and are expanded in vitro. Then, these tumor-infiltrating lymphocytes are transferred into patients and lead to tumor regression in some patients. T cells can also be genetically engineered to recognize tumor-associated antigens in various ways. That means the constructs containing specific T cell receptor genes, chimeric antigen receptor genes, or mouse T cell receptor genes could be transfected to T cells. And then, these genetically engineered T cells are transferred into patients. CAR T cells are the most popular engineered T cells. Chimeric antigen receptors, abbreviated as CAR, are also called artificial T cell receptors, chimeric T cell receptors, or chimeric immunoreceptors. They are engineered receptors which graft an arbitrary specificity onto an immune effector cell. The most common form of these molecules are fusions of single-chain variable fragments derived from monoclonal antibodies, which are fused to CD3 zeta transmembrane and cytoplasmic domain. Here is a flowchart for making CAR T cells. Briefly, T cells are isolated from patient blood, transfected with CAR genes, expanded in vitro, and then transferred back into patients. Nowadays, there are many clinical trials for CAR T in different kinds of tumor patients. The clinical data show that some of patients show significant response to this kind of therapy. Here is a patient named Mary Beth, who had non-Hodgkin lymphoma. In 2017, she received CAR T therapy. 1 month later, all tumors disappeared. But the cost is also high. Just like T cells, NK cells could also be isolated, expanded, or modified in vitro, and then transferred into patients and mediate strong anti-tumor responses. Similarly, NKT cells could also be isolated from a patient with cancer and expanded in vitro before being infused back into the patient. The second class of passive immunotherapy is the tumor-specific antibodies. These antibodies could directly mediate the anti-tumor response, or they could be conjugated with toxin or radioisotope to induce tumor cell killing directly. In summary, different types of tumor immunotherapies have been developed. In the future, personalized combined strategies would benefit the patients a lot. That's all for today's lecture. Thank you for your listening.